Hi everybody, welcome to Platinum Health. I'll be talking about the coronavirus or the disease that basically it caused, the COVID-19. Please share, subscribe, and like if you find it interesting. First, the name Corona. Where does that name basically comes from? It comes from the corona or the crown of the sun during eclipse. Under electronic microscope, the virus appears, as you can see in the pictures, as a sun during eclipse with the corona around it. So that's where the name comes from. I want to share with you some interesting facts about the virus. And basically, the virus is not a newly discovered one. In fact, it's been discovered since 1964. And it caused just a common cold. After that, the virus developed to cause a few epidemics, like SARS in 2004 and MERS in 2012, and a pandemic last year, and it is lasting until now. Hopefully, it will be gone soon. And all of these last three can cause severe respiratory syndrome. There are basically four more coronaviruses that cause just a common cold, which we could have been infected with at a certain point of our life. This is the one that they discovered in 1964, and this is an actual image that I was able to search for online. This is the SARS in 2004, and this is the MERS, the Middle Eastern Syndrome or Respiratory Syndrome in 2012. And this is the COVID-19. And as you can see, all these four, and even the other four that cause a common cold, they all have almost similar structure. So you can't see like that a huge difference, like maybe a little bit in, like nucleus or basically the inside of the virus. Otherwise, like they all have the spikes, they are kind of similar at all. Now, in order like for you to understand it, let's say in a better way, uh, I want to tell you about the structure of the virus. And don't worry, it's not gonna be boring, I promise. Uh, the main components, which are basically divided into two main categories, the structural proteins and the non-structural proteins. Also, there are accessory proteins. So this is a visual representation on where the structural and non-structural coding genes are on the viral RNA. You can see that basically this is when the virus basically enters the cell. It has this region, let's say this region of it. It goes or basically, sorry, it makes non-structural proteins. And these are the ones that make the structural proteins, and here the accessory proteins. This is another visual representation, and it shows where the structural components are. So you can see basically here we have the membrane protein, which I will talk about soon, the envelope protein, the famous spike glycoprotein, and I will say why it's called the glyco, and the lipid bilayer membrane. This is the spike protein. And basically spike protein is also, like as I said, it is a glycoprotein which is basically a carbohydrate and a protein. They are attached covalently, so they are attached strongly with each other, but the protein is the larger portion by mass. So let's say kind of by weight or mass, to be more, let's say, scientific. They are the contact points with our lung cells. In the spike protein is where one of the important mutations took place and made the spike protein fit better and more tightly than before with the receptors of our lung cells. Also, other mutations took place, of course, but this is one of the major ones. So basically, usually it's like here or here, here. It just makes the virus gets attached to our cells, or basically the receptor of our cells more tightly. This is the second protein, or the envelope protein, or proteins. They are present in small quantities, and their function is not entirely understood yet, but one of their functions is that they function as an ion channels. If you have, let's say, kind of a biology background, you know what basically these are, or basically they're ion channel. What are the ion channels? They just like allow the ions to flow from both directions. I think so that the RNA don't get damaged. This is the membrane protein. It's the most abundant on the virus's surface. Also, it induces the membrane's curvature and binds to the nucleic acid. And now you're going to wonder what's a nucleic acid. So the nucleic acid protein is also abundant. It binds to the viral RNA and protects it. As you can see over here, it keeps the RNA stable inside the virus. Mainly nucleic acid proteins link to each other in a long spiral way, so they wrap and coil the RNA. This way it is just more protected inside the virus. Now the non-structural proteins. There are too many non-structural proteins, but let's say in general, some of them, they are enzymes to make copies of the genome's RNA. Some of them slow down the infected cell's own protein production. Basically, our cells, they produce proteins for us to survive. So they slow down these protein productions, and they make the cell make proteins for their own. So to make, let's say, new coronaviruses. Some of them are just copy assistants. So they help the main, let's say, protein that makes the copies of the virus or the RNA of the virus, which is basically the coding sequence of the virus. It just helps that protein. Some of them are just like they camouflage the virus's genes so they don't get attacked. And some of them have proofreading activity, and this is just like, let's say, the influenza virus, it doesn't have a protein that have proofreading activity. That's what I think is most likely. But here we have proofreading activity protein. The NSP12, that's the protein that I was talking about. Basically, it adds it assembles, it makes copies of the gene's RNA. It assembles genetic letters into new viruses' genomes. It's this one. And the NSP7 and 8, these are, they just help the NSP12. They help this one. And actually, these, like these, they help this one. The red and the blue, they help the gray one. Now, if the NSP12 sometimes add a wrong letter to the new copy, the NSP14 cuts these errors and add the correct ones. It has proofreading activity. It makes sure that no wrong, let's say like the building blocks of DNA or RNA. Sometimes these machines, when they assemble, they are very accurate, but sometimes they make a mistake. They add a wrong building block in the RNA, and this one corrects it. And not all viruses have this. Now, the entry to the cell. Here I indicated the most important parts that are needed for the entry to the cell and replication, like the S protein over here, the genomic RNA, this one, and RNA polymerase, this one, and I have more pictures about it. And the RNA polymerase, yeah, I said this. And our cells, we have like too many proteins in reality over our cells, but these two are very important for, let's say, for our topic right now, which are basically the ACE2 receptor. This is the very important one, and this is very important one too, the serine protease. It's called serine protease. and it's also given the TN or this name, let's say TMPRSS2. And new research are still being done about this virus. It's still not known for 100% if there are more, let's say, receptor proteins or other proteins over here that are, let's say, playing an important part for the viruses entering into the cell. I read one of the articles and it says it's not only the ACE2. In fact, ACE2 is not that much, or the quantities are not a lot on the cell surface. So that article basically said there is another protein, and that protein is more abundant on the cell surface, and that one basically serve as a navigating system to until the virus gets attached to the ACE2 receptor. But that one's not clear yet. It's still like not. There's something like called in vitro and in vivo, and basically the research was being either done on the cell or on tubes. Let's say on the like more, let's say in a containers. So if you have like a test being done in a container or glass container, usually, as you know in chemistry, it's different than you do it on a cell or on the on the virus itself. So still, that one is not proven yet, but we know in all cases the ACE2 is the main component, or the one that gets the virus inside the cell. It's very important to know that the virus uses its own copy machine, or it's called also the RNA polymerase. It uses its own polymerase. And I also talked about it, as you remember, in the non-structural proteins. Also, the virus uses a positive strand of RNA. Now, you might wonder, what's a positive strand? The positive RNA is treated like mRNA. Now, what's mRNA? Basically, it can be directly read by the cellular, let's say there are machines, they are called ribosomes. These make proteins. So this positive RNA strand, it serves like an mRNA, which that mRNA it can be directly read. It doesn't have to be, let's say, converted to another one or another type or converted to negative so that it could be read. No, it just gets into the cell, and these ribosomes or the machines that make the proteins necessary for the virus, they directly start making proteins, and like the S proteins, the spike proteins. Now, unlike some viruses that need to enter the cell's nucleus in order to replicate, this one, no, it just gets into the cell surface as soon as it starts cleaving by the TMPRSS2. This one attaches it, and this one cleaves it. So as soon as it gets cleaved into the, let's say, the water of the cell or the cytoplasm, this ribosome comes in and starts making these proteins. So this is the attachment. As I told you, the ACE2 receptor, it binds to the S protein, and this one is the one that cleaves it into the cell. As you can see over here, it is cleaved. There are still, like, let's say, two theories about it. Some of them think it just gets into the cell and then gets cleaved. Some of them think while it is getting attached into the cell, or basically it's induced down, it gets cleaved. But in either way, when it cleaves, this is the RNA and this is the RNA polymerase. Don't confuse it with the ribosomes. Ribosomes are the ones that make proteins. This one that comes with the virus, this is this RNA polymerase. It just comes with the virus. As soon as it enters here, it starts attaching to this, and like here, it attaches to it and start making new copies. Now, as it is making new copies, the human ribosomes start also attaching to these strands and making proteins. It's also important to note that that basically there's kind of it can't just like start with anywhere on the RNA. The human ribosome, it has to start from a point where, and that point is called an open reading frame. Just think about it that it has a specific site to bind. For the human ribosome, it binds to a specific point, and that's when it starts making new proteins. And here I already explained this. Now, the translation of the virus protein or the viral proteins take place at different locations. So when it gets into the cell, or basically into the water of the cell, the whole process, it doesn't take place at like specific location or one specific location. I'm sorry, and it has let's say to go different places inside the cell. And for example, the spike proteins, the envelope proteins, and the membrane proteins, that the ones that all these four I talked about, all of these take place at something called ER, or the ER membrane. It's also called the endoplasmic reticulum. If you have background in biology, you know what that is. But basically, there is just like humans, we have organs. The cells also have organelles. So just like us having heart, we have kidneys. The cell have also things that it needs, let's say, to kind of survive, and in general they make us survive as well. But this one organelle is called endoplasmic reticulum, and these four proteins, the nucleic acid, spike, membrane, and envelope, they get basically translated over here. So they get made over here. Now the nucleic acid is translated in the cytoplasm. This is the nucleic acid, the one that we talked about, and we said it protects the RNA. It helps it coil, and it helps the RNA gets or stays in a safe place and intact. Now all of these proteins then get combined in another, let's say, complex. It's called an ER Golgi intermediate. So it is basically here. This is called the endoplasmic reticulum, as I said, and it goes to another thing called Golgi. So this is the one that gets between this and that one. Here they get mixed or assembled, as you can see here in the picture. And then there's something or kind of interactions called hydrophobic interaction. The hydrophobic interaction takes place, and then the exocytosis happens. And by the way, some of these pictures are mine, and some of these are not mine. Like those, they are not mine, but this is mine. I'm just saying this so you are clear about it. Now the treatments for COVID-19, we have hearing a lot of things about like this and that treatment. This is effective, this is not effective. But I grouped them here, or basically they are kind of grouped into groups that are based on their functions. Some of them block the entry, some of them block the replication, some of them innate immune response. They are just like, for example, like here in the blockade of entry, I talked about the anti-spike or the spike proteins. So some of them are anti-spike. So basically, it attacks the spike or the way the spike works. One of the most important ones is the remdesivir, which I think, almost like I would say 70% of people have heard about. Remdesivir, it blocks replication. The replication that I was just talking about. How it works is by RNA polymerase, the one that comes with the virus, that machine that I talked about, or that protein that comes with the virus. It grabs remdesivir, it grabs this medicine, and it attaches it to the newly made RNA, or the RNA chain. And we say chain because it's made of multiple nucleotides, so, or building blocks, let's say. So it adds it to this nucleotide. It basically confuses it with adenosine. So these nucleotides for RNA and DNA, they are four, but there's one difference between DNA and RNA. And one of them is called adenosine, and remdesivir is just, let's say, kind of similar to adenosine, and the RNA polymerase confuses it with adenosine. When this happens, the RNA becomes defective, it falls apart, and basically it stops, and the RNA polymerase or the RNA polymerase stops working from making more copies of the virus after just like few additions. There are other ones, and I found this one very interesting. It inhibits the TMPRSS2, which will not allow the virus to cleave to the cell. And when I'm saying like this, especially like this one, it is still not clear for 100%, but this is like, let's say, most likely what takes place. Regeneron. We all have heard about the Regeneron, and it is a therapeutic medication. And it is a mix of two antibodies, and some are derived from patients that have been treated from COVID-19. And these antibodies help the patient recover more quickly. Now for the vaccines. We all have heard about Pfizer and Moderna, and I'll explain about these. So how Pfizer works is by using messenger RNA, the mRNA, which I already I think introduced it, and I told you that this mRNA, as soon as it gets into the cell, the human ribosomes start, or these machines that make the proteins, they directly start working on it. And the reason behind that is because our cells make the RNA. But also scientists now can make them in labs, and Pfizer and Moderna, they are making this strand that codes only for the spike proteins. So I already told you that the virus uses or it has a genome that's positive strand of mRNA, and it's treated by our body as mRNA. The body then start making only spike proteins. So when the scientists make these mRNAs, they only make the part that makes the spike protein. So when they get into our cells, only the spike proteins are made. And this is a more clear picture. So these are the mRNAs that only make these red ones, the spike proteins. These get encapsulated, let's say, or coated with lipids, so they can attach more easily to the cell, and they get directly into the cell as well. And they bind to our lymph cells, not any type of cell. It's a lymph cell, because when they give the vaccine, they give it into they just inject it into our blood, and there are the lymph cells over there, and that's where these get attached. Then the immune system, after the spike proteins are made, the immune system responds to these spikes, and they promote antibodies and T cells, which basically just start attacking the virus if it gets into the cell. Now, one of the important things about this vaccine, in order to protect the mRNA, it must be stored in extremely low, -94 Fahrenheit temperature. For Moderna, it is like this storage temperature is one of the key differences between virus and Moderna, but between Pfizer and Moderna. Moderna can be stored in a fridge, The ideal temperature is -20, but it can still be effective if it's stored from 36 to 46 Fahrenheit for 30 days. There's also another vaccine. It's called Novavax, and that one uses like the simple. I'm just giving you the simple explanation of it. It basically uses the moth cells as a factory for these spike proteins, and these spike proteins get added with, let's say, other. It's called adjuvant, which is used to boost the immunity system to produce more antibodies and longer lasting immunity, and then it gets injected into the body. And then the body makes the also antibodies for this virus, and whenever the person then gets the COVID-19, we already then have the antibodies for it, so it shouldn't affect us, and that's how these vaccines work. Thank you very much for listening. Please subscribe, share, and comment if you have any questions. I couldn't go like or just like explain more in detail because it will take way longer time, and I know like now it's already 26 minutes. Probably some of you have already been skipping on the video, but that's, I felt like in order to understand the coronavirus, you have to know a lot of information about the structural proteins, non-structural proteins, and how it works. Basically, that's not how it works in detail. It's a little bit complicated, but thank you very much for watching, and again, like and subscribe and share. Thank you.